Manipal, and Professor Mandal from St. John Institute of Medicine from Bangalore. I have a great pleasure in welcoming and introducing the chairperson of this session, Professor A. Jagannatha Rao, who is a well-known scientist, and he is a reproductive biologist, more specifically reproductive endocrinologist. He has been working in the Indian Institute of Science, Bangalore, since more than 35 years with the responsible position. And presently, he is INSA senior scientist, and he was a recipient of Raja Ramanna Fellowship. And Professor Rao has contributed immensely to the field of reproductive biology. And he has received several national and international awards and recognitions from various organizations. Professor Rao is a good teacher, an excellent researcher, and a wonderful mentor who always supportive to young researchers and young scientific community. Sir, it gives me an immense pleasure in welcoming you to this Kolleg, and may I request you to take over the session. Thank you.
Dr. Ravindran, whom I have known for quite some time, is a product of JIPMA, one of the very famous institutions in India, and I am fortunate of having JIPMA students known as my PhD students to do extremely well during their career. He is currently, as I mentioned, now is a director of the Institute of Life Sciences at Bhubaneshwar, and he has is early training in JIPMA, as I mentioned, and later in Liverpool School of Tropical Medicine. And his main interest has been in the area of parasite biology. As you can see here now, there is a very interesting topic which he is going to discuss with us. His interests include immunological diseases, the evolution of the immune system, and over the years he has really built up the Institute of Life Sciences almost from scratch. There was a small building which I saw, now it's a very well nice building. For the years he has got 18 PhD students to participate, and 60 MD students, and he has published about 90 scientific papers. And I should add here that although the cardiovascular diseases as well as the other important diseases now attract attention, this is actually the parasitic diseases which are very rampant in India has attracted less attention. The only one which has really drawn worldwide attention for other reasons is malaria. But he is going to speak to us on nematodes and sepsis, relationship waiting to be harnessed. Dr. Ravindran.
All I need to do is to introduce you to three different, what appears to be little disconnected areas. I'll tell you what nematode biology is, and particularly in the context of lymphatic filariasis that we have been working on. Then I'll tell you about how exactly we got into research on sepsis. Basically, the investigations and the results drove us from nematodes into sepsis. And in the process, we also got to understand how the macrophage biology works in both these two diseases. The macrophage biology is something, an area that has been evolving over the last 10 years, although it's about a century since we knew about classical activation of macrophages from the time Elie Mechnikov discovered macrophages, but there is a whole group of macrophages which we have begun to understand what are called alternate activated macrophages. So we got into that area as a consequence of what we found. So I'll take you through that story slowly and steadily in the next 35-40 minutes, and then we'll have enough time to talk, question, and so on and so forth. So this is a life cycle of lymphatic filariasis. It's a nematode that is mosquito transmitted, and you get exposed to mosquito bites, and the infective larvae is a stage that comes, which sits on your skin and then buries itself and then goes into the lymphatics. For those of you who are not into the area of immunology, lymphatics essentially is the hub of immune activity, and here is a parasite which goes homes into the lymphatics, and it molds twice to become an adult as big as this. In the humans, the size of the worm can be as much as 3 centimeters, 2.5 to 3 centimeters, and the infective larvae that comes from the mosquito is about 1.5 millimeter, and it becomes an adult. And the adults, male and female, mate in the lymphatics and produce embryo in large numbers, what you see at the right bottom, microfilaria, which comes into the circulation to be taken up by the mosquitoes, and the cycle goes on. There are two things you need to remember, that there is no division of the bug inside the mammalian host, and basically the males and females reproduce, and that's how all nematodes reproduce and enhance their numbers. The duration of the stay of these parasites in the lymphatics can be as high as 15 to 20 years, and there are no drugs to treat them. All that you need to treat, if at all, an infected individual is to take diethyl carbamazine and a whole bunch of other drugs, which will eliminate the microfilaria from the circulation. But more important that you need to remember about this disease, it actually defies Koch's postulates. In a sense, an infected individual is largely without disease, and a diseased individual is largely free of infection. So the infection and disease are not necessarily happening in the same individual, and almost 90% of people who are diseased, those who have chronic pathology, are free of demonstrable infection. How does one demonstrate infection? There are multiple ways of doing it. At least there are two clear, definite ways of doing it. One, you look for microfilaria in circulation, and a far more sensitive method is to look for circulating filarial antigens, which you can score in plasma by a serological assay. Basically, the circulating filarial antigen that you get in peripheral blood in plasma is a product of the adult worm. It is constantly turning over, and a person who is infected, as I said, where the infective adult worms are present in lymphatics for 15 to 20 years, you will be able to demonstrate the presence of infection almost for 15 to 20 years. Sepsis is a completely different set of diseases, syndrome. Sepsis is a syndrome viewed as complex dysregulation of inflammation affecting multiple organs. Invading pathogens, very often bacteria, but there are viruses and fungi which will activate the immunological pathway and also coagulatory pathways and leads to septic shock and death. About 300,000 people die in US and about 550,000 people die only in the intensive care in Europe because of sepsis, and we do not know how many people die in this country of sepsis, which is a complication of bacterial and viral and sometimes fungal infections, because the number of people who die of terminal sepsis and shock is to be recorded only in intensive care hospitals, and the tertiary care hospitals, as you know, are not as many in this country for us to score. So it's not just in this country. As a matter of fact, whole of the developing world, we do not have actual picture of the number of people who go into sepsis and septic shock and die. So as I go along, I'm going to tie up these two different infections and diseases. The third thing that I want to orient you towards understanding what I'm going to talk about is a whole set of receptors, what are innate receptors that were discovered about 20 years ago, or what are called pattern recognition receptors, which recognize pathogen-associated molecular patterns that you see in different bugs, bacteria, viruses, fungi. And essentially, all these molecules that you see in bacteria, fungi, viruses, activate macrophages in a classical pathway to induce inflammation. And there is a direct link that sepsis is a consequence of this overactivation by bacteria, viruses, and fungi through pathogen-associated molecular patterns, activating pattern recognition receptors on macrophages. As I said, they can be of bacterial, viral, or fungal, or even protozoal origin. And excessive stimulation, as I said, leads to sepsis. We asked us this question a few years ago, that this list, for example, covers a whole variety of pathogens, but you don't see metazoan pathogens, you don't see nematodes here. So we asked this question, are there pattern recognition receptors to be discovered, actually yet to be discovered, we do not know at the moment, but this is what the search was for us to figure out if there are pattern recognition receptors that recognize metazoan pathogens. So the search for a metazoan specific innate receptor was the primary objective when we started all of this. This is just a cartoon demonstrating a whole variety of pattern recognition receptors. As you can see, there are different receptors what are broadly classified as toll-like receptors, which recognize specific patterns on different pathogens. For example, TLR2 recognizes lipoteichoic acid or peptidoglycan in gram-positive bacteria. TLR4 recognizes lipopolysaccharide in gram-negative bacteria. TLR5 recognizes flagellin that is present both in gram-positive or gram-negative bacteria. TLR9 recognizes CpG motifs that are widely present in viruses, and so on and so forth. So basically, the innate immune cells recognize these kind of patterns in pathogens and then get activated to an inflammation level. For those of you who are students, you need to recognize the inflammation that you see as a consequence of these bugs, and the innate immunity, the cellular and the molecular mechanisms are just about the same. And the underlying principles are the same, but the consequences, what you see as innate immunity and otherwise what you would witness as inflammation. So these are the pathways which have been figured out very extensively over the last 20 years in the different signaling pathways for all these TLR. I will not go into the details. Here is the initial set of findings that we found. So the whole thing is done by flow cytometry, and we are into a discovery of a receptor, unknown receptor on the surface of macrophages that recognize parasites. So we have labeled nematodes extracts of filarial parasites, and I will summarize the data here. There is a whole bunch of molecules of parasite origin of ill-defined nature. They bind to the surface of macrophages. They also bind to the surface of monocytes. So these parasite antigens need not necessarily come from filarial nematodes. You can also get it from C. elegans, which is a soil nematode. So it is a broad nematode specific molecule that is labeled and that gets bound to both mouse macrophages and human monocytes. They have no binding to lymphocytes. So this is a simple, straightforward cold binding, and which can be inhibited by unlabeled antigen. The next 1 year was spent in recognizing this, what this receptor is. Just to cut the long story short, I will tell you that this was recognized to be TLR4. I told you TLR4 was already recognized to be a receptor for lipopolysaccharide present in gram-negative bacteria, and lipopolysaccharide induces inflammation of macrophages, monocytes, both in all mammalian system. But here is a filarial nematode molecule that was binding to TLR4. And second thing, I need to remind you that lipopolysaccharide does not directly bind to TLR4 the way we have shown the filarial nematode component binding to TLR4. Lipopolysaccharide, on the other hand, binds to MD2 like LPS binding protein, and it's a whole complex that activates macrophages through TLR4. So the direct binding of LPS to TLR4 doesn't happen, whereas in this case this binds directly to TLR4. If it was not binding to TLR4, we would have even missed its binding. The next issue was what in the part of the parasite that binds to TLR4. Here again, a whole series of experiments were done. The bottom line was that it is a carbohydrate component that binds. It is not the protein part of the parasite that binds to TLR4 on the surface. So it is done by different variety of means. You treat with chitinase, the activity goes off. Chitin is because, as I told you, the microfilaria, the embryo, the last stage that I showed you, it has a thick sheath which is completely chitinous material. Nematodes are a very high rich in chitin. So the one that has much more chitin, probably, as you know, chitin is the second largest molecule nature makes because insect eggs, so the cuticle is full of pure chitin. The crab shell, for example, is the easiest and the best and purest source of chitin. So when you buy chitin from Sigma, it is nothing but crab shell washed, cleaned, and then powdered, insoluble, because it is N-acetyl D-glucosamine long chain cross-link and water insoluble, and that is what makes the crab shell hard, and that's what makes the insect egg cuticle hard enough. So that is chitin, and the same chitin is also present in nematode cuticle. It is also present in the eggshell of nematodes. It is present as a sheath on the microfilaria embryo. So we had reasons to assume that chitin possibly has a role to play in this binding when we found that it is primarily the carbohydrate part of the filarial glycoprotein that binds to TLR4. We can actually demonstrate that it is highly specific for TLR4. You can take cells and then overexpress TLR4 on the surface and demonstrate that it binds more. So the green histogram here is the overexpressed one, and the pink or the maroon is the steady state level, and here is the control. So simply by overexpressing TLR4 on a macrophage or on even Jurkat cells, you can demonstrate that the filarial glycoprotein binds much more. This is just the chemistry of the chitin that I talked to you about. Chitin is a long chain one, and if you deacetylate, you get what is called chitosan. Chitosan is nothing but poly-N-acetyl D-glucosamine, but the acetyl groups are all removed vastly, about 70 to 80%, and that makes it soluble. So when people talk about soluble chitin, very often what they talk about is chitosan, which is sort of deacetylated. But what we are going to be talking about is long chain synthetic sugars, which are poly-N-acetyl D-glucosamine with acetyl group intact, and you can have multiple units of it, residues of it. Chitobiose is 2 residues, chitotriose, tetros, pentos, hexose, and so on and so forth. You can go on lengthening the chain synthetically to make soluble chitin polymers. So what we demonstrated earlier was that the filarial glycoprotein that binds to TLR4 is essentially a carbohydrate, and this is a set of experiments by competitive inhibition in flow cytometry. We demonstrate that the longer the chain of N-acetyl D-glucosamine residues, the better it is in inhibiting the reaction of the filarial glycoprotein to TLR4, essentially saying that it is a sugar which is giving the binding, and the longer the chain, the better it binds to TLR4. It could also be demonstrated in silico, fortunately, because we have the crystal structure of TLR4 is known and published, and the 3D structure of chitohexose is also available, and it was a simple, straightforward bioinformatic demonstration that TLR4 binds to chitohexose. So what does this binding actually mean? We have to ask the question of biology. There is a filarial glycoprotein, and the sugar part of it has chitin polymer, and that binds to TLR4. So what does it do to the cells? So we go back to what was originally published, where you take macrophages and stimulate them with LPS, which was done about 20 years ago, and it activates macrophages, and a whole bunch of inflammatory molecules are made by the cells. So the set of inflammatory molecules that we look for are the prototype classical set of molecules, TNF alpha and IL-1 beta. You can also demonstrate IL-6, and since the pathway inducible nitric oxide synthase goes up, and that results in formation of nitric oxide, you can demonstrate as nitrate in the supernatant of activated cells. So the tall green bars are the expected results. That's what has been published and known, that when you take macrophages, murine macrophages or human monocytes, and stimulate them with LPS, lot of TNF, IL-1 beta, high nitrate and IL-6 are formed. This is classical information, widely known, published. A couple of people got Nobel Prize for demonstrating this and a variety of other genes associated with TLR4. And we demonstrated, here is a molecule, fallicyl antigen, or the sugar, which binds to TLR4, and we demonstrate that competitively it inhibits the activation of macrophages by lipopolysaccharide to induce inflammatory molecules. So this is to be expected. It appears to be sterically binding and inhibiting that activation of cells by lipopolysaccharide. This could be demonstrated in vitro, even for human monocytes. The one that I showed you, the previously was for the mouse macrophages. This is for human monocytes. The results are pretty much similar. I don't have to take you through this. LPS-mediated activation of human monocyte is inhibited by the sugar and also by fallicyl antigen. The whole set of protein expression could also be demonstrated by qPCR for inhibition of a whole variety of these genes, IL-1 beta, IL-6, TNF alpha, IL-12 and INOS. The open bars are the ones stimulated with LPS, and the cytohexose is the inhibition of LPS mediated, and these are obviously in the log scale. The inhibition is extremely significant. In the case of IL-1 beta, actually it is even suppressed, and even the steady state background levels are suppressed. And we also demonstrated that it has something to do with inhibition of ROS by cytohexose. So LPS alone will induce a lot of ROS, and in the presence of cytohexose, it gets inhibited. Can this be demonstrated in vivo? So far, what data I have shown you is that the cytohexose or the fallicyl antigen inhibits LPS-induced activation of cells in vitro, whether you take human monocytes or mouse macrophages. Can this be demonstrated in vivo? In vivo, the protocol is very simple, straightforward. This is again data demonstrated in literature. If you take LPS and inject in mouse, in 2 hours time, the kinetics is such that these molecules, TNF alpha, IL-1 beta, IL-6, peak at 2 hours, and then the levels go down over time. And this could be inhibited in vivo with sugar as well as the fallicyl antigen. So the inhibition that you see here for nitrate and for IL-6, IL-1 beta and TNF alpha. So here is what we have done so far, that we demonstrate a fallicyl glycoprotein that binds to the TLR4 receptor. It's a sugar, it is a cytohexose, and it inhibits activation of cells by LPS in vitro as well as in vivo in mouse. So the next logical question was, LPS-induced activation leads to sepsis, endotoxemia, and death of mice. Can this be blocked? The answer is yes. Whether you take BALB-C mice, which are far more susceptible to LPS-induced endotoxemia, or black 6 mice, which are relatively resistant, but nonetheless they are susceptible, except that the course of death takes much longer. In the case of BALB-C mouse, when you challenge with lethal dose of LPS, in less than 24 hours, you will see all the animals die. And when you inject this with fallicyl sugar, fallicyl antigen, or with a cytohexose, you block the death as a consequence. this death can be blocked even if you come and inject the sugar 24 hours after induction with the LPS. Even then it protects. So this is exactly what happens in sepsis. In a gram-negative sepsis, it's the LPS-induced activation of macrophages that results in sepsis. And in the case of humans, a sepsis patient essentially comes to the clinic at this stage when the clinical symptoms have already set in. So this experiment was done essentially to show that you can reverse not simply at the time of injecting with LPS, but you can come back even later and inhibit. So what I have shown you so far is that classical inhibition activation by LPS is blocked by the sugar. And is there anything else that the sugar itself does if it is inhibiting classical activation? What does it do after it binds to TLR4? We figured out that it actually activates the alternate pathway of macrophages. What is the signature molecules of alternate activation of pathway? As I said, the classical activation, which was known for almost several years, decades, is that they make TNF alpha, IL-1 beta, IL-6 and so on and so forth, which is the inflammatory pathway. The alternate or the pathway, the non-inflammatory pathway, is characterized in the case of mouse by macrophages making YM1 a transcription factor and arginase 1. This is the signature molecule. Simon Gordon in Oxford demonstrated about 2 decades ago the classification on features of the alternate activated macrophage pathway. So what actually does is that fallinergic antigen and sugar induces alternate activated pathway, and IL-10 and arginase, this is in human monocytes, this is in mouse macrophages. This is in mouse, YM1 and arginase 1. IL-10 is an anti-inflammatory molecule and arginase quantitative enzyme assay. This is in human. Here is a bit of a diversion, but at the same time which falls into the story. There are 2 strains of mice. C3H HEJ is a mouse which has a mutation in TLR4, and its wild type mouse is C3H OUJ. So if you take C3H HEJ mice and then stimulate with inject LPS, they will be resistant. This is a standard strain. Originally, this LPS resistant strain was a freak mutant in the NIH colony, which was characterized by Bill Paul, without knowing that it is TLR4, because the discovery of TLR4 happened almost 2 decades after the LPS resistant strain was discovered. So the molecular identity of LPS resistant strain that was discovered in the NIH colony was characterized 20 years later after the discovery of LPS receptor, which is TLR4. So we asked this question: if chitoic source or fallinergic antigen is binding to TLR4 and inhibiting and inducing alternate activated pathway, will this mutation make a difference for alternate activation? The mutation makes a big difference for the classical activation. None of the LPS induced classical activation will happen in the macrophages of the mutant strain, and what we have demonstrated effectively here is that the alternate activation is also dependent on that mutation. In other words, the mutation affects the alternate activated pathway by fallinergic antigen and sugar. We move to going back to the original disease model. Now we ask this question. Professor Rao, please tell me 5 minutes before time is due. Yeah, okay. So that I'll structure it. So we go back to human disease, and ask the question: will the fallinergic antigen bind to TLR4 of infected individuals? I told you in the beginning, the circulating fallinergic antigen is a marker of active infection in humans. So you can classify people in the endemic areas as those who have active infection because they will be circulating antigen positive, and those who do not have are the ones who do not have active infection. So when I say CFA positive, that means they are infected individuals, and CFA negative means they are uninfected individuals, or in fact individuals who have no active infection demonstrable. And then what we are doing here is binding of this antigen. What it shows us is that if you have an active infection, it's primarily resulting in lower binding of the filarial antigen to TLR4, assuming it is binding to TLR4, because this is an assay which is essentially only a binding assay. So it binds less, and our interpretation is it is because it is saturated in vivo with the same antigen that is present in circulation, and that is why it is inhibiting, which could be demonstrated. All you have to do is to take monocytes, put them in culture, allow it to recycle without the plasma of the individual with fetal carcinoma. The TLR4 recycles, and then the normal levels come back. You'll be able to demonstrate that the filarial antigen binds pretty much comparable to the normal individual simply by putting them in culture. All of this was done effectively to show that there is a constant saturation of TLR4 in infected individuals in vivo. And you need to remember, I said 15 to 20 years individuals are infected, so you are dealing with a situation where circulating filarial antigen is present for 15 to 20 years as long as the adult worms are in circulation. So this is a chronic maintenance of infection, chronic saturation of TLR4 in infected individuals by filarial antigen. And this saturation can be reversed simply by letting the receptor recycle. This is effectively the same thing as what I have said, that you can activate the cells, what I had shown earlier, that individuals who have circulating filarial antigen, you cannot activate them with titrated doses of LPS, but because they are saturated, you will not be able to activate them with LPS. But those who are free of infection, you can activate with LPS. You can demonstrate this for TNF alpha, IL-1 beta, or IL-6. So what exactly happened in vivo when you are challenging with LPS happens in vitro also in mouse, in humans, where individuals who are infected with filarial worms, where their TLR4 is constantly saturated, do not respond to LPS in vitro. I'll skip this. Oops. So we asked this question: if helminth glycoprotein or its carbohydrate units block experimental endotoxemia, are filariasis infected subjects resistant to sepsis? In other words, this is looking for an association after knowing the mechanism. We have demonstrated in mouse models, we have demonstrated in vitro that filarial infection leads to decreased stimulation with LPS. But will that mean that 15 to 20 years people live with infection, are they less susceptible to sepsis? It's hard to do this experiment. One of the ways of demonstrating is this. We take a large cohort of patients of sepsis in an ICU, and then we compare a large cohort of individuals who are having sepsis but without filarial infection. So we ask this question: if you take a cross section of patients with sepsis, how many of them would have circulating filarial antigen, an ongoing filarial infection as shown by circulating filarial antigen? And what is the prevalence of sepsis in patients with filarial antigen as opposed to those who do not have filarial antigen? But nonetheless, they all are in the hospital with sepsis. You don't have to see this busy slide. All that you need to focus yourself is in a normal, healthy control. When I say healthy, they are normal people in ICU for a variety of other purposes, trauma and other purposes, not filarial antigen positive. you have 42% of the individuals have ongoing filarial infection. This is pretty much the feature that you see in a variety of geographical area in filariasis endemic area. About 30 to 50% of the people will be infected. As I said, those who are infected are asymptomatic, so you will not know anyway. They don't have filarial disease, they will have infection, as opposed to only 6%. What it tells us is that an ongoing filarial infection leads to decreased prevalence of sepsis in a cross-sectional data in an ICU hospital. So, I will summarize here. This is, we did this work, we published this, but somebody else thought to make a model out of it. So Akim Horov from University of Bonn wrote a review recently where he has articulated what we had said, that cytoexos binds to TLR4 and leads to blocks alternate activation, and also it leads to alternate activated macrophages. He was trying to explain the filarial biology in a broad sense and how exactly this could be making a difference in decreasing sepsis. So I'll leave you with these slides. Most of this work was done by Santosh Panda, who has left the lab. The last bit of the work was done by Madhumita Panda, who has left the lab. And the one who is following it up on sepsis is Ratnadip Mukherjee, who is currently with us. Rick Mayfield at the University of Edinburgh and Eileen Devaney at the University of Glasgow have been my collaborators in the past, although their own contribution to this particular work was mostly in terms of giving me reagents and so on and so forth. Dr. Vidyut Das at the SCB Medical College is a clinical collaborator with whom I have a long-term association. He was my former PhD student. Vinita and Satyajit and Anna George have been my collaborators in a variety of other projects, and Tushar Vaidya at CCMB is another one with whom I collaborated in this work. Thank you very much, and I'll take questions. Inflammation that is associated with the disease and mortality is because of ongoing activation, for which LPS is already present in circulation in such humans as cytoexos. So as things being approached, I guess there is quite a bit. They had a back order and we could not buy for 9 months. So I'm simply assuming that a lot of people are buying. It's not terribly expensive and many people are trying out. So the approach is to take a not physical blockade. To answer your question, if somebody has severe sepsis and you are sitting amount of money at the moment, this is a Cephros column with polymyxin B. Polymyxin B has very high affinity, and you have to replace the cartridge about 15 to 20 times. Each cartridge, by Indian terms, it's about $1.5 lakh, and then the blood is transfused. There is nothing else to be done. The insurance has approved this as a mode of treatment, so patients don't care in the US. So this is what is being done at the moment, physically trying those. But experimental trials, clinical trials, there are the whole bunch of analogs out there. But 2 of the analogs reached the class phase 3 trial, and they've in a TLR4 knockout mouse, you cannot induce sepsis, but you can still get a little bit of LPS independent pathways of TLR4 independent pathway of.
This is Professor Amit Mandal, St. John's Research Institute, Bangalore. He is currently a professor of molecular medicine at clinical proteomics and clinical proteomics at St. John's Research Institute. He is basically told and has been extensively Mass Spectrometry for proteomics. This is an introduction from Hemoglobin to iron.
It's yours? No, this one.
And as a read, actually covered the internet.
Open all. Which one is yours? This one? I can see that. This one. Oh my God. Okay. This one? Yes. And he has received funding from the university to launch a proteomics project. And I think you hear from him what is exciting research he has been carrying out in the area of proteomics in Bangalore.
I would like to thank organizer for giving me opportunity, especially Professor Manipal, who invited me to talk on this platform. The title of the topic that I am going to talk about is hemoglobin structure function correlation in living human red blood cells using isotope exchange mass spectrometry. Before I describe you how did we monitor structure function relation of hemoglobin inside a live cell, let me take you quickly through isotope exchange based mass spectrometry. In proteins, the hydrogen atoms that are attached to or bonded to electronegative elements like nitrogen, oxygen, those are polar. And being polar, they are allowed to exchange, they are allowed to switch over its place from protein to aqueous solvents and back to protein again. And this phenomena is a continuous one. It's happening always in each and every cell in a biological system. Due to low electronegativity difference between carbon and hydrogen, CH bond is not polar, so those hydrogens are actually non-exchangeable. The hydrogens present as side chain residues in the amino acid in a peptide and at the terminal positions, they exchange at a very fast rate and it's difficult to detect. But those sitting at the peptide backbone, their exchange rates are relatively slow in the time scale of seconds, minutes, hours, and those are actually possible to monitor. Now, this exchange process can be catalyzed by acid as well as base. In acid catalyzed mechanism, there is transient protonation on the oxygen of carbonyl group of peptide bond, followed by removal of amide hydrogen in next step, which subsequently comes back, replaced back from solvent, and this phenomena goes on. Now, at this moment, under this condition, if instead of normal water, if we populate the medium with heavy water, D2O, instead of hydrogen, deuterium comes in, which actually adds 1 Dalton to its mass per exchange without changing chemical property, because deuterium is an isotope. And this exchange phenomena is highly dependent on pH and temperature. In large excess of D2O at a fixed pH and temperature, this HDX follows pseudo first order kinetics. The rate of HDX for different peptide amide hydrogen depends on their solvent accessibility, hydrogen bond strength, inductive and charge effect of neighboring groups, etc. So, monitoring this event using mass spectrometry is basically defined as isotope exchange based mass spectrometry. As I said that it is highly dependent on pH, so the rate of HD exchange for peptide amide hydrogen drops with the decrease in pH, and it becomes minimum at pH 3, where acid and base catalyzed rates are equal. So, generally HDX is performed under physiological condition, physiological pH, where the proteins are in their native state, and then to follow the exchange kinetics, the exchanged proteins is quenched rather, by decreasing temperature to 0 degree and dropping the pH at 3, and subsequently that protein is proteolysed using pepsin under quenched condition, and ultimately we generate a set of peptides which we analyze in mass spec, where exchange information in the peptide is retained back. So, say for a particular peptide, if it has n number of exchangeable hydrogens, the number of deuterium incorporated at a particular time can be calculated through this equation, where Mt and M0 and M infinity are the masses of that peptide at time t, at 0 time and infinite time. That means when all exchange phenomena has been accomplished. Now, in a peptide with n amide hydrogens, exchangeable amide hydrogens, principally each of those contribute distinctly. So dt can be expressed as a summation of n exponential terms. But in real life, this equation can be simplified. Those amide hydrogens can be grouped into three major categories: fast exchanging, intermediate, and slow exchanging ones. So dt can be written as a summation of three exponential terms, where A, B, and C are the number of fast exchanging amide hydrogen, intermediate one, and slowest one, and K1, K2, K3 are their respective rate constants. So literally what we need to do, the moment we ask a question that how protein conformation is changing with a particular biological event, we need to monitor this exchange kinetics of protein in its initial state and in final state, and then generate set of peptides and analyze them, and then by the plot of dt against t, we should have to calculate these different kinetic parameters A, B, C, populations, and rate constants. And then basically we have 6 kinetic parameters for each peptide in a particular state. So moment we ask the question that how is conformation changing, we have to deal with 6 kinetic parameters of a particular peptide originated from protein in its initial state and in its final state. So basically we have to deal with 12 kinetic parameters, which is a bit complex situation. So we have simplified the job by using the method of initial rate calculation. In, as per chemical kinetics, the rate of initial rate of any reaction can be written as rate constant multiplied by the initial concentration of reactants. So for a particular category of exchangeable amide hydrogens, I mean to say fast exchanging, all we need to calculate is K, a product of K and A, population and rate constant, and then take the difference of that particular group across 2 states, initial and final states, and then take the sum of these differential rates across all peptides, which ultimately tells us how the conformation is changing, whether it is becoming more flexible or it's becoming rigid. So sign of this algebraic summation will tell us some idea. Means a positive sign gives the idea that yes, at that portion in the protein during the biological event, flexibility has increased, and a negative sign says decreased. So basically, a biological event can be traced across the polypeptide backbone of a protein using HDX-MS. So now to get into insights of any biological events, basically we need to monitor the structure function correlation of protein within cell. But there is a difficulty. We know that all structural biologists actually deal with purified proteins. Say, given an example, say if we need to monitor intrinsic fluorescence property of protein, and if we have a cell, if we shine light and try to analyze emergent beam from the cell, emergent fluorescent beam, it's very difficult. It's absolutely impossible to distinguish the contribution of thousands of different proteins in that emergent fluorescence beam. So the routine task for a structural biologist is to purify protein, analyze the structure in vitro, and then extend that information in vivo to correlate with its function. But the ground reality is cellular complexity is very high. Each molecule is sitting with thousands of others, and in vitro and in vivo environments are radically different. So the question remains unanswered: how to study molecular structure and dynamics inside the living cell? We hope that isotope exchange based mass spectrometry might be the answer. I am going to take you through a couple of slides where I guess I'd be able to convince you that yes, this technology has the answer. So basically here I am going to present the structure function correlation of hemoglobin executed through HDX, monitored through HDX when exchange process executed inside the living cell. So we have taken a very simple strategy. See, water is permeable across any cell membrane, and D2O, D being isotope of hydrogen, D2O should also be. So we allowed RBC to incubate in D2O containing buffer, maintaining all physiological condition. D2O was allowed to go inside, and then exchange was allowed to happen inside the living cell, living RBC. And time kinetics of exchange process was monitored by quenching at different time points, followed by proteolytic digestion using pepsin. And then those fragments originated from hemoglobin, exchanged hemoglobin, were studied in mass spec. Now, shift in this envelope with time across higher M by Z scale actually gives the information of all those kinetic parameters that I talked about from the plot of dt versus t. Let me give you the rationale why we have chosen hemoglobin and RBC. When we realized that yes, it's possible to visualize a molecule inside cell, we started through this HDX MS, we started looking whether somebody has reported already. To our knowledge, so far I haven't seen any report in cell HDX phenomena has been used to monitor the structure function relation of a protein. So we felt that before telling any mechanism, better we establish the method. So we have to go in opposite direction rather, like we have to choose a candidate where in vivo and in vitro environments are not very different, and for that RBC is the ideal candidate because RBC's hemoglobin content is 95% of total protein. So basically RBC you can consider as a in vivo system of pure hemoglobin. And secondly, we have to choose a candidate, a protein candidate where structure function correlation has been very well established, and for that hemoglobin is also the best candidate, I believe. For last couple of decades, people have worked a lot extensively on this structure function relation. So we have taken RBC, chosen hemoglobin as a candidate molecule, studied in vivo exchange kinetics, got information on oxygenation process, and then actually correlated, tried to correlate those in vivo observation with in vitro reports. I'll show you a few of the peptides. See, hemoglobin is a tetrameric molecule consisting of two subunit, globin subunits in duplicate, alpha and beta, and each of them are carrying one heme unit with where it is basically porphyrin nucleus, and iron is sitting at the center. Now, this is a part of hemoglobin in its deoxygenated state, ligand unbound state, and this is basically, this line is showing the plane of porphyrin nucleus, and at the center iron is there. In deoxyhemoglobin, the spin state of iron 2 plus is it is in high spin state, and the radius is such that it cannot fit at the center of porphyrin nucleus, it is out of the plane. And this iron is basically covalently bound to a histidine residue known as defined as proximal histidine residue in this F helix. Moment it is oxygenated, it binds to oxygen. Moment iron binds to oxygen, then it changes, it switches over its spin state from high to low, and that causes its reduction in its radius, and as a result iron is pulled into the center of porphyrin nucleus, and while doing so actually it pulls its covalently attached histidine residue along with it, and as a result there is this pocket between helix F and H. This size of this pocket reduces, and at that pocket one residue, tyrosine 145 residue at the C terminal end, that resides, but reduction in this pocket size causes tyrosine to expelled out. So, a tyrosine cannot reside anymore here, and while doing so, it actually breaks many interactions, hydrogen bond, salt bridge and all. And actually the function of hemoglobin is regulated by cooperative ligand binding and allosteric factors. Those are the things guiding its function, and those factors are regulated by structural transition. That's why the structure function correlation is very important for it. Now, in our HDX study, we observed a peptide, proteolytic peptide generated from exchanged hemoglobin 1921 from beta chain spanning residues 86 to 102, where we observed that kinetic parameters in cell HDX kinetic parameters showed that this region of the molecule on oxygenation is becoming flexible. Now, let us see what are the in vitro report says. One of the residue, valine 98, this one in this experimental peptide, it was bounded with tyrosine 145. This tyrosine, moment it is expelled out, that hydrogen bond dissociates. Again, salt bridge with the next residue between ASP 94 and histidine 146, that is also broken. And obviously, moment these interactions are disappearing, the exchange rate across the peptide in this region of the molecule is expected to increase, the region becoming flexible. Other interactions like say hydrogen bond between ASP 99 and alpha tyrosine 42, ASP 99 alpha ASN 97 are weakened. So disappearance of that interaction also tells that the portion should be flexible. But another hydrogen bond between ASN 102, which is the last residue here, and ASP alpha ASP 94, that means at the interfacial region, that is formed in the oxystate, and obviously that will lead to increase in rigidity. So if we take all interactions together, it is obvious that as a net effect, there is overall increase in flexibility in this region of the molecule, which is what we have observed in our in vivo study as well. So at least it shows that our in vivo result correlates well with the in vitro information. I have talked about allosteric factors that are important in functional assay of hemoglobin. One of them is DPG, 2,3-diphosphoglycerate. It binds in the pocket between two beta residues in deoxystate. Moment hemoglobin is oxygenated, this DPG is out from the molecule because the pocket size reduces, and moment it goes out, all residues which are involved in keeping DPG there, that becomes free. So obviously one can expect that in that region flexibility should increase on oxygenation. So we observed a peptide 1494 from beta globin chain, again consisting residues 1 to 14, where we have seen that yes, flexibility is increasing on oxygenation. Now coming back to this diagram, this is DPG. The two of the residues that are associated in DPG binding is valine 1 and histidine 2, which are these two residues. So these two residues are becoming free on oxygenation, and obviously it should contribute towards increase in flexibility. But there is another bond happening, like forming GLU7, this glutamic acid getting associated with beta lysine 132 in the oxystate, and that obviously imparts rigidity. So this may be the association could be the cause of relatively lower increase in flexibility for this peptide. Other DPG bound residues. How am I doing with time, sir? Okay, fine. Okay. Other DPG binding residues like histidine 143, lysine 82. Now, histidine 143 containing residues we observed in this peptide 1869. For this particular peptide, we have seen very low change in flexibility. It's difficult to conclude. It's very little flexible. Though we know that on oxygenation DPG is out from the molecule, so it literally makes this histidine free. So let us see what's going on in that region. What are the other residues? See, this histidine, the 146 histidine, was associated with aspartic acid 94 and alpha lysine 14 deoxystate. On oxygenation, as I explained previously, this tyrosine is out, so this it pulls this histidine as well, and then these two bonding interactions disappear. So we do expect that maybe histidine should contribute towards flexibility, but that's not the life. Histidine becomes associated with another residue, alpha lysine 144 of the same peptide, and obviously that puts much structural constraint within same peptide itself, and instead of flexibility, rather it imparts rigidity. The other interactions like on oxygenation following salt bridges are formed, which obviously imparts rigidity, and as I explained previously, this tyrosine residue which is thrown away, so all interactions with it in the deoxy disappear, so it contributes towards flexibility. So if we see this particular peptide, there are many flexible rigid interactions, and we calculate this value as a net sum, and obviously there is a balance between all interactions, disappearing interaction and reappearing interactions in this particular peptide, which perhaps the cause of very low flexibility that we have observed in this peptide. So in this way, we have analyzed 14 different peptides from hemoglobin, and we were able to achieve the sequence coverage of alpha globin chain by 55.8% and that of beta globin chain by 68%. We have seen many peptides. For many peptides, we have calculated the flexibility, rigidity informations from our kinetic model and HDX. But in many of those cases, which may be functionally important, but there was lack of sufficient structural information, in vitro information to correlate with. So, what we observed is in-cell HDX data corroborated well with the structure function correlation of hemoglobin established in vitro. Here we have used molecular mass as a probe, which is unique to the molecule. So this strategy can be used to study the structure of a molecule even in a unpurified system, means when it is sitting with other molecules without being purified. Using HDXMS, biological events can be seen at the molecular level in vivo, in cell. Whatever I presented is overall phenomena, what's happening in the peptide, but if you really interested to see one particular amide exchange, which particular amide exchange is changing in which way, then you have to come down to residue level, and for that purpose HDXMS should be coupled to tandem mass spectrometry. Generally 90% cases or even more for tandem mass spectrometry, the people use the strategy of collision induced dissociation, but other reports where HDX phenomena has been studied in vitro, people have seen that if the moment they use MSMS using CID, they lose the information because with the available energy, the exchange information is lost, deuterium hops from one place to the other. And people have seen that if they change the strategy, electron capture dissociation or electron transfer dissociation, then it's possible to retain back that information. But unfortunately, I don't have the facility of ECD and ATD, so I didn't able to do that. A mass spec being highly sensitive technique, so it can monitor molecular phenomena at very low concentration, which is very important for cellular entity to observe. For a complex environment, like in this case we have studied hemoglobin, which is abundant protein, we didn't have difficulty to see it. But in case of cellular, like any cell, if you are interested for low abundant protein, then you need to have a fractionation step added in front of a mass analyzer. So liquid chromatography coupled to HDMX would be better choice in those cases, and this LC coupled HDMX module is commercially available nowadays. Ion mobility, which separates the molecule in space on the basis of their shape, that can also be added to add another dimension for the separation, which would be again useful for low abundant proteins to see. And finally, HDX-MS is applicable to any protein in cell irrespective of its location and structural complexity. See, we have used HDX phenomena, which is there for decades. People are using specifically in enema for a couple of decades, hydrogen deuterium exchange. But those studies are in vitro studies with purified molecule. Moment you use the relatively newer technology, soft ionization based mass spectrometry, it's possible to go inside the cell. And obviously, you can use these strategies to see n number of biological events inside the cell where the main biology is. So I would conclude the talk with the quote from famous physicist, Professor Freeman Dyson, that science is often driven by new technologies rather than new concepts. Thank you. Thank you for listening to me.
Thank you very much for your illuminating talk. I just want to make 2 points in this session. Dr. Girisha, who is a professor at Kasturba Medical College, Manipal, in MD in pediatrics and MD in medical genetics.
Just check it out. Thank you.
Good morning. Thanks for this nice introduction, and I thank Dr. Devnath Pal and Dr. Satish Adiga for inviting me to be here. I am going to discuss our work on one of the rare genetic diseases which we have selected for our study. This study has been done over last 4 years. I bring warm wishes from Manipal to all of you. Adiga and Professor Pratap Kumar are representing our institute in this meeting. And this entire work is funded by Indian Council of Medical Research. We completed this project last year, and the entire work is done by one of my PhD students. He is about to submit it in this year. Mucopolysaccharidosis are a group of genetic disorders. We have around 9 mucopolysaccharidosis depending on the enzymatic defect. They are the complex biochemical molecules where if there is a defect in metabolizing these molecules or degradation of these molecules, then we get substances accumulated at various stages. What we are interested in is Morquio syndrome A and Morquio syndrome B, which are also called MPS 4A and 4B. MPS stands for mucopolysaccharidosis, and Morquio A and Morquio B are the syndromes. These two syndromes result from abnormal degradation of keratan sulfate, which is a glycosaminoglycan. It is a very complex structure which I will not be able to explain, but two enzymes, N-acetyl galactosamine 6-sulfatase and beta-galactosidase. When they are deficient, we get Morquio syndrome. So, in the same metabolic pathway, there are other diseases, Sanfilippo D syndrome and GM2 gangliosidosis. This is one of the glycosaminoglycans. There are several of them, chondroitin sulfate, heparan sulfate, and so on. So, what we will be dealing is only Morquio syndrome with two types. In fact, clinically they are indistinguishable, only biochemically they have different enzyme deficiency, but both result in accumulation of keratan sulfate. If we see the clinical features, all of them result from accumulation of keratan sulfate. If it gets accumulated in the liver, which it does, we get hepatomegaly or enlarged liver size. In the heart, it causes valvular heart disease, sometimes cardiac failure as well. Wide skeletal manifestations, corneal clouding. Though vision is not affected, cornea appear hazy, and widely spaced teeth, hearing loss. These are the common features, but skeleton is maximum affected to a maximum extent. You can see here, this girl has short trunk and limbs are relatively well preserved, but you can see the pectus carinatum, that is prominent chest, and here you can see the spines, vertebral bodies are very thin. We call it platyspondyly, and there is beaking of the vertebra anteriorly. So, this is the very classical feature of Morquio syndrome. And in the hands we get abnormal bones, they have wrist joint laxity. So, they are very loose and we can easily move across the wrist and the tone is less, and hands are slightly abnormal. You can see this is because of the abnormal bony changes here at the wrist. And the most dangerous complication of this disease is cervical spine instability. Due to trivial incidents, they can damage their spinal cord, which often can result in loss of control over all the limbs or sometimes even death. And they can have abnormalities in any bone, including the knee. So, this is a condition which we have studied, and as a rule we don't have incidence of these rare diseases in India, but in other countries also it is mainly an estimate. So, it may affect about 1 in 1 lakh to 1 in 5 lakh population in different countries, and India we do not have population based studies to know the incidence of this condition. Galactose, this GALNS is one gene which is the most common cause of Morquio syndrome, has 14 exons and has a signal peptide and sulfatase region in the protein. So, this is located on chromosome number 16q. And when we began the study in 2010, we had 153 mutations reported. Most of them were missense, spread across the gene, and few of them were splicing, some are small indels, some small insertions were also there. Gross insertions and complex rearrangements were also reported. Most of the techniques do not identify mutations in the intronic region, which we also encountered in our study. And the second gene is beta-galactosidase, which has 16 exons. Very few cases have been reported to occur because of the mutations in second gene. In fact, we did not get any patient with deficiency of beta-galactosidase causing Morquio B syndrome. We do not know why it is so, but I have asked all our Indian colleagues, they have not seen Morquio syndrome B, though in the literature we have about 20 to 25 cases reported. So, 21 missense mutations and 2 small insertions were reported in Morquio syndrome B in the literature. The objectives of our study were to analyze the clinical profile of Morquio syndrome and to identify mutations in GALNS gene in Indian patients with Morquio syndrome. The second gene we did not get any case. To study the functional consequences of novel mutations associated with the disease phenotype. We obtained the approval of ethics committee for this, and then obtained clinical and radiological features of Morquio syndrome from various centers across our country. We had in fact collaborators from 18 centers for this work, and blood samples were obtained from patients, parents and healthy controls. Those who had normal values for both enzymes were excluded from the study. So, this is a study design. Basically, we did enzyme assay to confirm the diagnosis, then extracted DNA from the leukocytes, and then we sequenced the entire gene in all patients. and then we carried out STR analysis and then bioinformatic analysis to see whether determine their pathogenicity, and sequenced healthy controls and looked into the databases to determine which is a mutation and which is a polymorphism. So coming to the result, we recruited 100 subjects as of now, and we could screen 86 of them, and because of poor quality of the DNA we could not perform any assay on 6 of them, and sequencing is in progress for 8 patients. So we identified out of 86 subjects, we identified both the mutation. This is an autosomal recessive condition, so every patient should have 2 mutations. In 83 individuals, in one we could identify only 1 mutation, and we could not identify any mutation in 2 subjects, suggesting that they are probably in the non-coding region or in the intronic region, which we are not able to identify during by our sequencing method. We have analyzed phenotypes for 78 patients. Some data were missing for some patients, but we could see here pectus carinatum, genu valgum, lumbar kyphosis, joint laxity. So these are the most common symptoms, and radiologically the anterior beaking of thoracolumbar vertebrae is the most common finding, which makes us diagnose this condition, and platyspondyly. So these are all the classical clinical features, and you could see how they are represented in our patients group. While studying these patients, we recognized clinically 2 subsets of patients, one with mild manifestation, one with severe. So you can see here, this is a child with mild severe manifestation. On the right hand you have severe manifestation. You can see here in the wrist. So this is almost well preserved wrist, and here you can see how disorganized the wrist is. In the pelvis, this is a hip joint. You can see here how, this is very severe disease suggesting destruction of the hip joint, and here again you can see mild destruction but not as severe as this. We could clearly distinguish these 2 phenotypes clinically, and we have 6 patients with mild phenotype, so we are really not able to make a genotype and phenotype correlation for mild versus severe disease. Again in the spine you can see here the anterior beaking. This appears like a bird's beak. You can imagine which bird it represents. There are several birds, and you can see here this is very mild manifestation of the disease in the spine. So this is the spectrum of mutation we found in our patients. So we got 46 mutations in the entire gene in our total patients, that is 86 patients, where we could complete the study, and we could see here. So IN stands for India. So we could see some are confined to our population, some are shared with other populations as well. So most of them were missense, very few of them are splicing or deletions or small insertions. Okay, so this is the continuation of the same list, which lists all the mutations which we found in our study. If we see the distribution across the gene, almost all the exons are involved in patients, and we can see that distribution, this is like any other population which is being studied. Mutations can occur in any exon or any location in the gene. It is not something like achondroplasia or sickle cell disease, where the mutations are localized to only one exon or only one nucleotide region. In fact, if you take the example of sickle cell, unlike that, it just demonstrates genetic heterogeneity. Within the same gene, different alleles may be there to cause the disease. We then performed in silico analysis by using several bioinformatics tools, mutation tester, Panther, Polyphen, SIFT. These are the standard tools, and in addition we did this through one more Indian software, that is Hansa, developed by Dr. Nagarajaram, who was one of the co-investigators for this study, collaborators for this study, and all the mutations were verified to be pathogenic by bioinformatic tools. Basically, when we begin the study, we have clinical features and enzyme assay which shows or which proves that they have the disease. We just have to find out the mutation. So it is partly easy because we have a low enzyme activity which is already proven, or where we already have a functional analysis done for these patients. And Dr. Nagarajan did the bioinformatic analysis. I am not really good in this, but he showed that all the missense mutations cause poor enzyme activity or decreased enzyme activity because of the involvement of catalytic sites of this particular enzyme. So this is X-ray structure of human GLNS gene, and you can see here several. This is another way of representing how or analyzing how mutations can cause poor activity of the enzyme. And when we saw the allele frequency, so we could see few of the alleles were common in Indian population. You can see here in exon 1, exon 7 and 8, majority of the mutations were concentrated in our Indian population. So mutant alleles in exon 1, 7 and 8 account for nearly 40 to 50% of the mutation. We really want to save money when we test the patient. So instead of sequencing all the 14 exons, this may be one of the good strategies to sequence only these 3 exons to begin with. This is what we are doing when we have request for diagnosis now. This is a very cost effective strategy. You save more than 50% of the expenses when we test this. This was possible because of our study, and instead of 14 exons you test 3 exons and you get mutation in nearly 50% of the patient. Very interestingly, we have observed something different as well. I think this is expected because of our geographic distribution. We had collaborators from Hyderabad, Kochi, Lucknow and Delhi, and we could say that if patient is from Hyderabad region, these are the mutations which are likely to occur, and from Kerala these are the 3 common mutations, and in Lucknow and Delhi these were the common mutations. We had one patient from Pakistan, and the patient had the same mutation as the patients have in Lucknow. You can go back to the independence or pre-independence era when there was a migration. So they shared the same mutation. So this is very easily explained. So probably we have not studied this aspect in our other diseases. We have different spectrum of mutation confined to different geographic locations. So that again will help us to save money on testing these patients in a particular region. So my student now tells, if the patient is from Hyderabad, I will test this mutation first. If the patient is from Kerala, I will probably test these mutations first. Once we complete our research, when we do it for diagnosis, then probably that is going to be cost effective, and you know the particular region has well defined mutations. We probably will expand this into other diseases which we are studying currently and see whether this is true for other diseases as well, which we expect it to be. Because if it has shown this trend for one disease, it is very likely to show for other rare diseases as well. We just presented our data on polymorphisms in this gene. Whenever when we sequence the gene, we find several sequence variations which are not pathogenic, but it is very important to provide this kind of data for other groups which are working on the same disease, that these are polymorphism. We really have a different pathogenic mutation, so that interpretation becomes easier for some other scientists. So in fact, this is part of our publication as well, polymorphisms in Indian patients which are not reported. So we have, I think, 29 new polymorphisms being reported in this gene in Indian population. To summarize, we had identified 25 novel mutations in Indian population out of, I think it is more than 200 now, because there were several other groups which were working simultaneously on this disease, and every day you get new mutations from different geographic region, but our study could contribute 25 new mutations. Most of them were missense, 3 of them splicing, 2 frame shift and 1 nonsense mutation in the literature. And these were the 21 reported mutations were identified in our population, suggesting that Indians really have a different spectrum of mutation. They have the same disease, but the mutation spectrum is different for different population. This is widely as per the expected lines. The work is now accepted for publication in American Journal of Medical Genetics. The proof is corrected, and I think it should be online within few days. So you can see here the team of people, I think 28 people from 18 different institutes have contributed to this work. When we submitted this work, this was the largest study on Markov syndrome and first from India. But now we have another publication by by American and European collaboration by a diagnostic laboratory. They have published larger than ours, but when we submitted, this was the largest study. The advantage of being in India is that for any genetic disease, we have the largest number of people or largest number of patients for research. So we can easily score over all other our American and European counterparts in terms of number, and that is what we have done in this disease. This entire work was possible because of my students and colleagues. In fact, Abdul is the one who is pursuing his PhD in this subject. Thank you very much. Controls are normal, healthy individuals. To have the disease, it should be less than 2% or 3% of the normal activity. All right. 0%, 1%. In fact, if we have more than 5%, they don't have the disease. That is the concept of enzyme.
Manipal is a very enthusiastic clinician, interested in basic research and established a very nourishing IVF center at the Manipal Medical College, and he is a member of the World Committee of Medical Issues of Ultrasound. Also as the National Chairman of Imaging Sciences and Obstetrics and Gynecology of India. He is also the leader of the Committee of British Medical Journal and several other memberships. And he is going to speak to us on translating experimental approach to clinical practice to improve infertility success. Was also here the assisted reproductive technology, which is a fast-growing science.
Thank you, sir, for the nice introduction. I must thank the organizers for having invited me for this lovely meeting with several scientists and some of us clinicians. I'm a clinician, so I was just discussing with Professor Rao how there are so many things to be done in translating things into practice. Once again, greetings from Manipal University. It's a university building with 4 floors of library for students and 4 floors of administrative wing. Now, if you're looking at the small video, you'll understand that the ovary basically produces a lot of oocytes. Now, in clinical practice, what one should understand is, in an in vitro fertilization program, we always generate a lot of oocytes by stimulating with gonadotrophins. So, when an ovulation occurs, normally in the body, the ovum gets into the fallopian tube of the woman and it waits for the sperms to get into the tube and the fertilization occurs there. It's a very basic concept of normal physiological process of fertilization. So now what one needs to understand is, in an assisted reproductive technology, what do we do in clinical practice and how experiments and research can go on connected to the basic physiological process. So in vitro fertilization is a concept where we try to fertilize the sperm and the egg outside the body, which everybody knows. But what do we do as clinicians is, we always know that normally only one oocyte is produced or comes out in a body, but in an assisted reproductive technology, we really get to know a lot of gonadotrophins are to be given for increasing the number of oocytes. So that is the beauty of the whole system of in vitro fertilization, wherein we create a lot of eggs. We do ultrasound guided procedures like that into the woman. We put the probe into the vagina, we put the needle through that vagina and we remove the oocytes from the ovary, which is a technical skills are necessary for a clinician to do this, because there are a lot of vital structures around the ovary, because sometimes there can be complications with this kind of technique. However, the oocytes are removed from the ovary and basically they are fertilized outside the fallopian tube of the body. So we know that this is basically indicated for the oligoasteroteratospermia, O80 syndrome, means we know that the sperms are low in number. When they're low in number, I always say it's a God curse. The God's curse is when somebody has a low number, they have a low motility, and they also have a very poor genetic potentials. So when we know that the simple techniques like intrauterine inseminations clinically are done to improve fertility, if it fails, we have to go in for assisted reproductive technology. And we also have a lot of endometriosis, means the women menstruate outside uterus and all this blood gets collected in the peritoneal cavity, and it's called endometriosis. Lots and lots of indication. However, a lot of men are born without sperms also, and that's called as azoospermia. We have over the years realized that a man who has no sperms can still produce a baby, but not all of them, because sometimes there can be obstruction for the sperm to come out, or there can be non-obstructive. So what we do clinically is, if a man has a very small testis and a high follicle stimulating hormone, he doesn't have sperms. So the other group is taken into assisted reproductive technology. We take a biopsy of the testis and we can store it in the laboratory for future use. These are the different technology. So assisted reproductive technology is just a simulation of the human body in the laboratory for fertilization. As I just mentioned, normally a lot of oocytes are recruited in the body for a one single ovum, and most of them are gone. Apoptosis occurs. I was just talking to Professor Rao. Can you all scientists think of something, how one can retain these oocytes in the body for a woman rather than just allowing it to be going off into apoptosis process? Nobody has thought of it. I think the translational theory and experiments can be on this, because we are finding a lot of women getting into premature menopause by 35 years. And we know maybe in the future science and the scientists can bring up these kind of experiments. Why is it going away? And can we retain those oocytes in the body? And that's what we do. In IVF cycle, we produce a lot of them, and we help those oocytes to come up, and basically they get fertilized. So what we do is, we have two protocols clinically. I don't think it's necessary for me to explain at the moment, but we all know in normal physiological process, there's something called as LH surge. Just before ovulation, the surge comes up like that, and the oocytes go away. And what we have to do in the clinical practice is, we will control that LH surge. So LH surge is controlled by two mechanisms, called a long protocol and short protocol. But basically we do kind of put a stop by giving gonadotropin releasing hormones. So when you do these kind of hormone analogs, the LH surge is suppressed. So as a clinician, we want a mature oocyte to come up and not the LH surges, and the oocyte sometimes get lost. So we have a lot of protocol called antagonist protocol. Basically we antagonize the LH by kind of manipulating the ovarian cycle. But I personally feel these have been standardized, and there is no big doubt about that. So what's the big doubt about IVF is, later on we know that though we can aspirate the oocytes like this, and I'm just showing a small clipping as to how these oocytes are being picked up by the follicle, and you know that this oocyte is inside the follicle there. We put a needle, as I showed on the small video clipping, and this needle will aspirate the fluids, and this is basically looked into under the microscope, and we know that how many oocytes are got by this manipulative technique of oocyte aspiration. But our main aim is, of course, to have a good embryo, and these are all the different equipment needed for our embryo. I think the science has understood till there how to get good embryos. I think we have understood that. But the next process is, whatever equipments have been brought in, we have also understood if somebody has very low sperm count, what do? We can inject under these kind of intracytoplasmic sperm injection, and we are doing Professor Satish Adiga, who is the coordinator for the scientific session, is with us, and he is doing this intracytoplasmic sperm injection. And we know that, I always tell, the woman is born with the largest cell in the body, and the man is born with the smallest cell of the body, the sperm. The largest cell in the woman is the egg. This all the assisted reproductive technology deals